thank you so much for the invitation. I would like to thank actually Rusin. We have known for many years now actually, and thanks to her, she introduced me to I think like the M&M conference series, and then since then we have been communicate a lot, and I also want to thank the UltraPath organizers for the opportunity to share my research on developing some interesting imaging technique that might one day be able to use in the pathology lab. And so, by the way, I am actually faculty members of biological science at the Carnegie Mellon University. And just a disclaimer, I'm also a co-founder of a startup called Magnify Bioscience, dedicated to distribute some of the kits that leverage the technologies that I'm going to talk about today. Okay, so now I'm going to just jump in. So the title of the talk is Making Super Resolution Practical, Magnify Expansion Microscopy for Research and Translational Applications. So first, before we dive in, I would like to have a quick one-on-one for this expansion microscopy technique, because I think for the audience, this might be some new technique. So expansion microscopy is a super resolution technique that only emerged, I think, roughly 11 years ago. Basically from the Professor Ed Boylan's lab at MIT. Initially, the motivation of developing expansion microscopy is to resolve the brain. Basically, try to image the the circuitry of the brain, because brain is incredibly complicated. It has billions of neurons forming trillions of connections, and those connections we call synapse, which is only like 600 nanometers, size objects, and it also packed with thousands of biomolecules of different kinds. So in reality, this is really hard to image. For example, if you try to use optical microscopy to image it, it will look like this, pixelated patches, right? It's really due to the physical diffraction limits. What I mean is, like, because light microscopy use light, so therefore the resolution really limited by the wavelength of light. So anything that's smaller than 250 to 300 nanometers, it's basically cannot be resolved by the optical microscopy. Of course, people develop super resolution optical imaging technique, for example, like SOM, STAT, and among others, that can circumvent this diffraction limited. But those techniques are oftentimes expensive and also require complicated sample preparation. So I think that one of the message, I guess, from these stories is that getting the nanoscale information from the complex biological system are costly and also slow, right? And here just shows you the range of the resolution for various microscopy, for example, electron microscopy, that allows you to image, sub-nanoscale information, but it's expensive compared to the light microscopy, while light microscopy, it's limited by the diffraction limits in terms of the resolution. Now, the question is, can we actually think of out-of-box idea to overcome, to fill the gap of resolution? And that's where Professor Boylan coined with the concept of expansion microscopy. Here, I'm just going to use the Earth as example. It's an analogy, right? So we treat the Earth as like a sample, and then you use a conventional optical confocal microscopy to image, let's say, a building or cells, right? In this case, it's Mellon Institute. That's where I am sitting at right now talking. As you can see on the left panels, due to the resolution limits, you would not be able to see the detail of the building. However, if you imagine we can actually expand the Earth several times in each dimension, using the same microscope, now you will be able to see the expanded melon institute, and you can see the expanded trees and cars, and much more details regarding the buildings. So that is basically the concept, right? To chemically transform the biological sample, embed them into a hydrogel, and then be able to like make the sample actually bigger in size. And here just shows you one of the video. It's a human breast biopsy tissue that embedded in a hydrogel that are water soluble, and then you throw them in the water. You can see it literally expanding in front of you, and you can actually see the gland structure because I immunostain the breast tissue. So since the invention of expansion microscopy, we really developed various of different techniques, and nowadays there are more than probably 100 different kinds, and but you can sum them up into 4 basic steps. First, you have to chemically modify the biomolecule of the tissue, and then you need to synthesize the water-soluble polymers in situ. And after that, you have to apply either enzymes or heat denaturants to denature the proteins. That allows the sample can expandable in the water. There are a bunch of review articles right here showing, and we recently also published one of the most recent one in Nature Review Method Primers. If you're interested, you can look it up. So here, I just want to show you one of the animation, just give you a concrete idea, right? So first, we apply the small chemicals to modify the tissue biomolecule. This is chemical, just like chemical handles that allows these biomolecules forming covalent bonds with the monomer that we are used to synthesize the hydrogel network within the tissue. So as you can see that like these small little balls are the monomers, and they are forming this gigantic hydrogel polymer networks. And during this process, the modified tissue molecules are now connected to the network. And after that, you can apply enzymes or heat denaturation to loosen up the tissue. And then you place the tissue gel inside the pure water. Now, everything are expanding, and these biomolecules, they are separate from each other while maintaining the relative shape and the geometry relationship relative to each other. Since I was a postdoc with Professor Eboyden at MIT like roughly 10 years ago, I already been working on translating expansion microscopy and for pathological application. And actually, my first work on expansion microscopy is to develop a technique like that. It's called expansion pathology. The idea is that we try to convert the clinical samples into expandable formats. So there are multiple different kinds of clinical sample, for example, like FFPE, H&E, and also like fresh frozen. So we try to convert them into a state that allows you to execute expansion microscopy procedure. And we also implement enhanced enzymatic digestion steps that digest away the extracellular matrices of the sample that provide the mechanic strands. So with this approach, you will be able to physically expand any clinical sample. For example, in this case, there's human kidney samples on the slide, and this is the left side of the photo, and then you can see that like after expansion, it looks like this, right? It's a transparent gel on the petri dish. So why we want to do expansion pathology? For instance, we use this to detect some of the kidney diseases that oftentimes require clinical grade electron microscopy to image, right? So I'm not going to go through this anatomy stuff, but I think one of the disease called kidney minimum chain disease that has the kidney polycyte food processing effacement. As you can see here, right, this is the EM images of the cross-section of the polycyte food processes, and it's continuous, while in the normal kidneys, you will see the periodic structure of this cross-section of the food processes. And the size is roughly about 200 nanometers apart, right? And this is actually beyond the optical resolution limits that offered by the optical microscope. So now the question is whether you can expand the kidney biopsy and then be able to see those structures. And here's the answer, right? So you have these normal kidneys that are expanded, and then we can immunostain with a bunch of markers, for example, vimentin, actinin-4, and collagen. And they can see that the actinin-4 actually show up enriched in the kidney polycyte regions. And then if you actually draw a line and line profile, you will see the periodic structure with the distance similar to what you observe in the EM. While in the case of the kidney minimum change disease, you do the same thing, expand them using expansion pathology by about 4 times. And then now you can see when you draw the line profile, you see that literally the periodic structure disappear, just like what EM show. We even try to collect these images and then run a single blinded experiment. So we have some volunteers to look at the expansion pathology images of the kidney, basically give them the ground truth, let them learn about it, and then show them the unlabeled images. And then they are able to correctly point out which one has the minimum change disease with accuracy 90% compared to the one that provided by regular fluorescent images. So expansion microscopy field has been rapidly evolving, and there are many different methods. So after I finished my postdoc work with Professor Boyden at MIT, I have my independent labs. One of the things I want to overcome is that the classical expansion microscopy method actually has certain features that are not suitable for clinical application. For example, it takes days to finish the process, and the gel oftentimes are very fragile, not very user-friendly. And also the lack of a universal anchoring mechanism. What I mean is, if you want to image RNA, you will have to apply a very different chemicals. If you want to image lipids, you will have to apply different chemicals. So you have to shop around the literature to figure out what kind of chemistry that are suitable for those target biomolecules for expansion microscopy application. And for regular biologists and pathologists, this is not ideal. And not to mention, the expansion factor are too small. So, only 4 times, that provide you roughly 70 nanometers resolution. That's okay, but not quite enough, for many applications. So, with those in mind, we would like to really develop a technology that are easy enough to use and universally applicable to different kinds of sample, and as well as different kinds of biomolecule. And even more so that even my son can actually do the experiment, right? So the answer is Magnify expansion microscopy. So we developed that a couple of years ago. There's some, advancement with this Magnify technology. So first of all, we developed a universal anchoring molecule. It's called methacrolane, that are compatible with all other, gel monomers components, so they can mix together. Therefore, we essentially eliminate the anchoring steps that and cut off the time by a couple hours. And not only that, we also modify the gel formula so that the Magnify gel, compared to other expansion microscopy gel, can expand up to 11 times, depending on your tissue type. So I'm going to go through quickly some of the features of Magnify. First of all, Magnify is a really a one-day process compared to the several days process of the traditional methods. And it's very simple. For example, you have a tissue, you place that on the glass slides, you submerge them with the gelling solution. The anchoring and the gelling will finish at the same time. So after 1 hour's gelation, you get the gel, and then you cut it out and place them in the tube. with the, we call that softening solution, and then you put it in the pressure cooker. After 1.5 hours, the tissue already processed, and then you can proceed with washing and staining, with your antibodies and other label agent. Then you will be able to expand the sample and then ready for imaging. The whole process only lasts for several hours. If you start it at the morning, you will finish by the end of the day. Because we use universal molecular anchors, so we can actually retain all kinds of biomolecule, proteins, lipids, nucleic acids. So here just show some example. For example, we retain the protein very efficiently compared to the old methods using agar-ags. And then the retention of the protein remain efficient across different types of organs and tissues. We retain lipids, so that means that you can actually use traditional lipid stain after expansion. For example, DII, DID. In this case, this is actually a presynaptic compartment. The yellow is the synaptophysin markers that highlight the synaptic vesicles. And then this purple thing is actually the mitochondria lipids. So we use a regular lipid stain to see them. So you can actually see the pristine structure because we expand this 11 times. So the resolution is sufficient to resolve these pristine structure of mitochondria. And then also because we retain the nucleic acid, so you can perform, for example, RNA imaging. You can perform DNA fish. Of course, you can also stain the nuclei using DAPI. With this, you can do something interesting. For example, you can image clusters of extracellular matrices. In this case, it's in a human stem cell derived lung organoids. So on the left side, this is the EM images, right? So you can see the clusters of the EVs on the EM. But then now with the Magnify, not only you can see them, but also you can actually establish the 3D reconstruction of the clusters. Not only that, you can even optically section the EVs, so you can actually see the complex structures inside those EVs. Because we expand 11 times, if you actually apply NHS ester fluorophore, you can actually stain literally all the proteins that preserve in hydrogel that give you a fake EM texture. So in this case, for example, we're showing this presynaptic dense projection. Panel 4 is the EM images of the presynaptic dense projection. You see there's some 3 blobs. We can also observe the same thing, 1, 2, 3, right? You can see that in the Magnify images. Not only that, because it's a 3-dimension, so you can actually rotate the images and be able to see this hexagonal structure that are really hard to find using electron microscopy technique. Of course, with the resolution, I'm talking about 20 to 30 nanometers. After 11 times expansion, you will be able to quantify the distance between the pair proteins across different brain regions in a relatively fast, fresh fashion. And also, if you combine with some computational approach, you will even be able to boost the resolution a little bit further, push it into a 15 nanometers regime. And in that case, you will be able to see, for example, ciliary structure. Like on the left side, this is the EM images of the cilia from the same lung organoid that I just talked about. And then consider in the middle, using the Magnify, you will be able to see the same cilia. We can see the microtubule bundles, the nifold symmetry, as well as the centroid area with the structure. Of course, we don't have sufficient resolution to resolve the internal structure of individual microtubule bundles, but at least you will be able to count the numbers of the microtubule. And then, not to mention, you will be able to establish a 3D structure of these ciliary structures. So this is just an extreme. You can spend about 1 hour to acquire a stack like this using Magnify approach, and then be able to generate this intact ciliary machinery. Right? So you can see the basal bodies, like the appendage, as well as the intact ciliary structure in 3 dimensions. The whole process only lasts for 1 hour compared to serial block phase SEM technique that acquire the same volume would take you weeks, if not months. We then also asked like whether we can use Magnify expansion to actually detect some ciliopathy. So, for example, here, you have the normal cilial structure. You can count. There's a 9-fold here, and then the abnormal one, they're oftentimes missing one of them, one of the microtubule structures. So we actually examined 3 different conditions. One is lung organoid divided from patient with PCD. Another one is a normal organoid that treated with Taxol, which is a cancer drug that can, we know that will damage the tubular growth of the cilia. And then, of course, we have the normal organoid. And as you can see, we try to do the same counting of the defective cilia among the whole population. And then the EM method and the Magnify method statistically are identical. So that means that they are equally effective for accurately quantify the changes related to those conditions. And Magnify expansion, unlike other expansion method, can actually expand a wide range of samples, including some of the tough samples, for example, kidney as well as like cornea. So I'm just showing you some of the examples, like kidney, prostate, before and after expansion, and the distortion is very small computationally. And we can expand all kinds of different tissue from different organs. I mean, one of the good things about using this Magnify expansion microscopy approach is that not only you take advantages of the conventional optical microscope, which is operating at a very fast speed, you can image the whole tissue even in the expanded state, and then you can switch to the high magnification, dive in, and then be able to image the nanoscale structure. For example, in this case, directly visualize the kidney food processes while still be able to do immunofluorescence staining so that you can get the molecular contrast that you need to tell the difference between different markers. So in this case, I just want to showcase some of the ongoing study using Magnify expansion microscopy. So my student are actually collaborating with local hospital and the Guinea General Hospital. We have 20 kidney patients with 10 disease. And then we are now using Magnify, coupled with a bunch of different immunomarkers to see if we can establish a workflow to use Magnify to diagnose kidney disease without the need of electron microscopy. On the left, you can see this is the electron microscopy images of the kidney glomeruli structure. And then on the right, we do the same, but using Magnify. Not only that we can have the EM texture, but also we will be able to get molecular contrast, for example, provided by antibody against ACTN4. We can apply lectin to highlight, for example, the basement membrane as well. And of course, we can see the nuclei using DAPI. We developed software to automatically segment out those markers. So we can, for example, automatically detect basement membrane. We can also detect the food processes and then be able to quantify the result automatically. With this approach, we can detect a bunch of different kidney disease without the electron microscopy. So, for instance, like this is normal versus membranes, glomerulus nephritis. So, using this quantified approach, we were able to measure the thickness of the glomerular membrane, basement membrane thickness. And as you can see that the disease state has a thicker thickness compared to the normal state, which is only like 300 nanometers thickness. And of course, you can also detect some of the interesting nanostructure that normally requires electron microscopy to see. For example, in this case, the zebra bodies, in the fibroid diseases. So we can actually image those, see those bubbles, without the need of electron microscopy, using only confocal microscope. I mentioned the eye infection. So in that case, we actually developed a variant. It's called MicroMagnify that combine Magnify process with a cocktail of enzyme that digests away the cell walls of the pathogens. This approach is pretty, broadly applicable to different kinds of pathogens, including gram-negative, gram-positive bacteria, as well as fungus. With this approach, we received sample from Wills Eye Hospital in PA, and then we got those samples. Those are the human cornea samples that infected with all kinds of different pathogens, and then we are able to expand all of them, and then use different stain to highlight the location of the pathogen. For example, here, this is C. albicans infected human cornea. So you can see the yellow, those are the fungus. And then we can also expand other human cornea that infected by various different pathogens. For example, like mycobacteria. For example, like this amoeba. So yeah, and then because Magnify preserve the epitopes, so we can actually image many colors if you want, because like the markers are actually stay inside the hydrogel. So you can apply the immunostain and then you remove them, and then you stain a different set of labels and then do this again and again. So I hope that I just show you evidence that Magnify enables enhanced super resolution imaging on the pathological samples using light microscopy. We would like to distribute this technology as broadly as possible. So we do provide DIY information so that people can try to repeat those experiments inside the lab. We also try to test whether people ultimate test is actually to commercialize it. So we also provide easy to use kits from the startup. So far we already have more than 300 labs and biotech company all over the world using this technology. We are also trying to push development of bright field based Magnify expansion microscopy. So in this case, actually everything that you saw is acquired from the bright field microscope. So that means that in the near future, we'll be able to use slide scanner to actually image those nanoscale features. For example, in this case, the food processes as highlighted in this black arrows, using without the need of electron microscopy. We also try to develop AI models. Due to the time, I'm going to skip fast. So we try to collect bladder cancer, muscle invasive bladder cancer patients' biopsies. We stain with 4 different markers: DAPI, WGA, TLC, as well as the centromere. And after that, we collect those tissue images, and then we feed them into the AI models that we develop. And then we try to do a comparison. So it turns out that Magnify allows basically review some of the nanoscale features that allows the machine learning models to accurately predict the treatment outcome of those muscle invasive bladder cancer patients. And here's the comparison with the same machine learning model. If you let it train on the H&E, we can get roughly 79% accuracy for telling which patient are going to benefit from the treatment, which one are not. However, if with Magnify approach, the additional information allows the model to boost the accuracy by about 12%. And then one of the amazing thing about this approach is that the AI model will be able to tell us where to look at. So we will be able to actually study on the tissue level as well as on the single cell level which patches or which region we should pay attention to. This is something that ongoing, and we are actually writing up the manuscript, so stay tuned on this development. And with that, I would like to stop here. Thank you so much for attention, and I'm happy to take any question at the end of this section. Thank you.